Hello and welcome to the third lecture in this ongoing series of lectures on chemistry in everyday life. In today's lecture, we will study about certain diseases and drugs which are used for those diseases based upon our knowledge which we have accrued in the first two lectures about receptors, enzyme and mechanism of drugs. So, we will rather apply that knowledge in today's lecture. We will talk about in this lecture about antacids, antihistamines, analgesics, tranquilizers, antibiotics, antiseptics and disinfectants. So, we will straight away move to the first, antacids. Antacids, to know about antacids, one must also appreciate the fact that the pH of our stomach is highly acidic. It is around 1.5 to 2.5 when empty, and it raises to 5 to 6 when food is ingested. The acid which it contains is hydrochloric acid. Then what happens is, well, acid needs to be secreted when food enters the body, okay. But under certain diseased conditions, this acid can actually accumulate in stomach even when food is not to be ingested. It can happen under the following conditions, like if somebody is consuming alcohol, if somebody is anxious, eating certain kind of foods like primarily junk foods tend to release a lot more amount of HCl than required. Smoking, then certain drugs as a side effect of those drugs like aspirin and all others NSAIDs, non-inflammatory inflammatory non-steroidal drugs, and then it can also happen because of somebody's stressful conditions. Now, the problem is, once this unwarranted acid accumulates in stomach, it starts to corrode the epithelial lining of the neighboring tissues, for example, gastric epithelium, and then it creates ulcers like small wounds, right. So, it can create gastric ulcers in stomach, it can create duodenum ulcers, it can create esophagus ulcer, or in general it can create gastritis, which is inflammation of the gastric mucosa, which is that epithelia lining the stomach cavity. So, it can get in general it can get inflamed. So, which means that excessive release of HCl is a pathogenic condition, right. And since it is an acid, one most natural antacid which might come to our mind as chemists would be addition of a base, right. So, that a neutralization reaction would ensue with addition of gastric acid to the base we are talking about, and that would result in formation of a buffer. So, that should be the the most basic kind of antacid available through this drug design maneuvers, and of course, this is the case. So, one can find a lot of such antacids, a lot of such bases in the market, and based upon the nature of those antacids, we can have systematic antacids, for example, sodium bicarbonate. Now, when we say something as systematic antacid, what do we mean by that? Generally, all these antacids which fall in this category of systematic antacids are highly water soluble. So, when a salt is very highly water soluble, what it is going to do? It is going to be readily absorbed in bloodstream, right. And as soon as it gets into the bloodstream, now it has no role to do in bloodstream, right, because it is going to neutralize acid which is present in stomach, not in bloodstream. But as and when it gets into the bloodstream, it tends to give a lot of metal ions, and then those metal ions starts to accumulate systematically in the blood circulation, which if goes unchecked for a prolonged use of such antacids, it can lead to a stage where blood plasma becomes highly alkaline, and that is a very toxic condition for a human being, right. So, that is what we mean by systematic antacids. When we talk of non-systematic antacids, we talk of certain salts, certain compounds, basic compounds, which are not that soluble in water. Now, if they are not that soluble in water, they will remain poorly absorbed in the bloodstream. So, then this kind of alkaline condition of blood plasma would not probably exist, and hence these are much safer in comparison to systematic antacids. Antacids falling in this category include salts of aluminum, salts of magnesium, salts of calcium, and you can go and check in a pharmaceutical store, all these are available on counter. You can just for the sake of curiosity, you can just go and have a look what kind of salts are being used as antacids there. Apart from all that, there are also certain miscellaneous antacids, something of the kind alginates, which are again which are polymeric compounds in nature, they are polymeric sugars, or like samethicone, which are again polymeric compounds, something of the kind of some polymeric silicas. But primarily when we talk of antacids, we talk of generally those metal salts. Now, if one is to look for an ideal antacid, there are certain properties which an ideal antacid must possess. It should exert effect rapidly and over a long period of time. Then reaction of an antacid with hydrochloric acid should not cause large evolution of gas, should not be constipative or laxative, right. Should make a buffer in the range of 4 to 6 pH. It should not be absorber or cause systemic alkalosis, something of the kind I talked about in the previous slide when I talked about sodium bicarbonate. Then it should also not interfere with absorption of food, and last but not the least, it should be inexpensive. Well, these are too many characteristics to be kind of expected in an antacid, and as expected, unfortunately, none of the antacids available so far would meet all these criteria mentioned in the previous slide, and every antacid would have one side effect or the other, especially when it is used for a longer period of time. For example, when we talk of sodium bicarbonate, number one, it is a systematic antacid, and secondly, it tends to also release carbon dioxide in stomach. So, it has limitations on 2 counts. Similarly, because you have a lot of accumulation of sodium over a prolonged period, it can also lead to heart failures. So, for patients which are on low sodium diet, they are never fed on antacids which are of this kind. Aluminum hydroxide, it is a good antacid, but it causes constipation. Likewise, when we talk of magnesium salts, they are having an opposite effect, they act as laxative. Okay? Whereas calcium carbonate, upon prolonged uses, it can cause hypercalcemia, so which means a lot of calcium accumulation in the body. Now, sometimes to kind of counteract, to circumvent these side effects, one method which chemists use is they give you a cocktail or a mixture of more than two components. For example, magnesium and aluminum salts can be formulated in one antacid. So, what is going to happen? Magnesium salts, they are supposed to be faster acting, whereas aluminum salts, they are supposed to be long lasting as far as antacid is concerned. Likewise, I told you magnesium salts are generally laxatives, whereas aluminum salts, they cause constipation. So, again kind of they both compensate for each other's deficiencies and drawbacks. Now, there is another kind of anti-ulcer compound, and those compounds are antihistamines. These compounds work through an entirely different mechanism, okay, and that's very there is a very interesting chemistry around antihistamines as far as anti-ulcer activity is concerned. Before then, we must understand histamines. So, histamine is this structure shown on your screens. It is an imidazole scaffold which is joined to an amine through an ethylenic bridge, and this amine is in quaternarized state. It's in a protonated state under normal plasma pH. Now, what this compound does? Generally, this compound increases permeability of blood vessels. It's actually a hormone. It can also be neurotransmitter at times, based upon the action we are talking about. So, I spoke about the action of hormones or neurotransmitters, and I told you in the previous lecture that these are nothing but messenger molecules, and these messenger molecules then fit on certain receptors, and based upon their binding on those receptors, a certain pharmaceutical effect would be produced, right. So, it means it has to bind to certain receptor. Now, what that receptor is going to do? Once it fits into that receptor opens up and lays down a cascade of activities which eventually lead to increased permeability of blood vessels. It also activates immune system. Now, both these activities are important when, let us say, some pathogen has entered the body, right. For example, if somebody has got certain viral infections, let us say it is somebody is suffering from cold. So, then a lot of this histamine activity would be incited, and the impact, physiological impact of this histamine, this activity would be that a person would exhibit symptoms as seen during cold, right. So, you would see that runny nose, you would see redness of nose. It is all because of increased permeability of blood vessels, and then you have, since your immune system is activated, so it tends to wash off all those viral particles and all. The major limitation is that it can also happen when somebody is allergic to certain things, like for example dust or pollens. So, the body is not able to differentiate between a virus and a dust particle, and the same phenomena can actually be exerted, and a person can get all the same symptoms of, and something which happens during hay fever and all. So, for which there is a class of compounds which we call antihistamines. Now, why I am talking about this, which has no relevance to the ulcer and antacids and all which we are studying. Scientists somewhere down the line realized that histamine is also responsible for production of HCl, okay, and production of large amount of HCl. Now, when they started to delve into that, what they found was that these are the parietal cells which are actually manning this stomach cavity. So, on these cells generally are present certain receptors, and those receptors are able to receive neurotransmitter in the form of acetylcholine and hormones like gastrin and histamines. So, what happens when all the three compounds, for example, they bind to their respective receptors present on the parietal cell? Due to their action, they act as an agonists, and they incite this parietal cell to release a lot of amount of HCl, which means that antihistamines can actually act as also antacid compounds, right. But there was a catch. So, scientists thereafter realized that there are two entirely different kinds of receptors, H1 receptors and H2 receptors. Both are histamine receptors. Okay. So, the important thing scientists figured out was there were two kinds of receptors. Both these receptors acted upon histamines. One was H1 receptors and another one they found was H2 receptors. Now, H1 receptors, they were histamine receptors basically involved in inflammation process, something of the kind I told you in 2 slides before this slide, where I spoke about hay fever and all. Now, the second type of receptors, which was this H2 receptor, they were responsible for gastric acid secretion, and it is this H2 receptor which was of use to scientists in order to develop an anti-ulcer compound. So, now the second question was, since this histamine was an agonist for this receptor, we must be able to find out an antagonist for this particular receptor. So, what it meant was we must effectively convert the agonist, which was histamine, into another compound which will act as an antagonist. Now, there is this very important point where we know what is an agonist and we tend to develop an antagonist primarily based upon a platform which agonist provides us. Okay. To understand this, I will provide you an hypothetical situation. Let us assume that here lies a receptor, and this receptor is acting as a gatekeeper for this ion channel. Okay. Now, there is a compound, this compound here, which acts as an agonist of this particular receptor. Now, by being an agonist, it means that as soon as this agonist binds to the receptor binding site, due to induced fit mechanism which I spoke about in the previous lecture, there would be certain conformational changes in the receptor shape also. And because of that change in the receptor, you see this gatekeeper, this gate has now opened up because of agonist binding to it. What does it mean? It means that probably what has happened is that this agonist has been able to bind initially to the active site, but the binding was not very strong, and there were still certain more prospective areas of binding, let us say here, which were not really ideally conformationally designed or placed. So, in order to kind of reinforce this binding, there has to have certain structural changes in the receptor. Now, by being changed into this more constricted shape, it has brought effectively this carboxylic group quite in proximal distance to this protonated amine, and then this ionic bond has been forged. But because as soon as this interaction has taken place, it has also resulted in opening up of this ion channel. That is what an agonist is going to do. Now, we are interested in developing an antagonist, right. So, what we should do? We should try to design a molecule, number one, that it should probably be similar to the agonist, but it should be designed in such a way that it is able to forge much better interactions with the binding cavity, in such a manner that in that case this receptor does not have to change its shape, like what has been happening here. So, as soon as this antagonist fits into this pocket, okay, number one, it fits into this pocket much better than the agonist. So, even if there is some agonist present, antagonist will always be preferred in this binding site. Number two, it fits it in such a manner that there is no change in the receptor, and then this ion channel, that ion channel which we spoke about, remain closed, right. So, then what we see is that antagonists generally are better fit than agonist molecules. Now, keeping the same philosophy in mind, we will try to evaluate how histamine binds in its active site. Again, this is more hypothetical, this is more cartoonish, but there are similar binding sites in actuality. So, if we look at histamine molecule and its binding into its active H2 receptor site, we find that there are two different kind of binding possible. First is through this protonated amine, and that can engage through ionic interactions with the binding site in the active site. Now, from this amine there is this flexible 2 carbon chain, okay, which leads to this particular imidazole molecule. In this imidazole molecule, what we see is this imidine unit, this amidine part, NH, CH and double bond N, and this amidine unit can engage in other hydrogen bonding interactions with the active site. Okay. So, basically these are the interactions we are looking at when we are thinking of histamine in its active site. So, now let us picturize this whole thing in context to development of an antagonist. Let us say if this histamine molecule, as soon as it fits into the active site, it changes this particular active site through induced fit mechanism, and which means that there is certain space it leaves here, right. It has changed its shape from this to this. Now, that would mean that if we are able to add to certain steric crowd here, for example, let us say steric crowd or certain extra functionality. Right now we do not know which functionalities will be actually good, but believe that there are certain extra functionalities. If we are able to get to this extra functionality on histamine molecule, then this molecule will become rather a better fit in comparison to histamine, and then this receptor would not have to change shape. And if that does not happen, it means we would have developed a perfect antagonist. So, keeping this in mind, starting from histamine, people were able to design a lot of compounds, lot of H2 antagonist compounds, for example, methiamide, for example, cimetidine, for example, ranitidine, and other compounds also, and all had been hugely successful molecules. As far as methiamide is concerned, you know what we have changed. So, here instead of having now quaternary amine, we have replaced it with this kind of thiourea bond, right. The problem with this compound is that patients who suffer from kidney damage, they are not susceptible to this molecule. Next one in line was cimetidine. Now, in this case cimetidine, the basic changes, we have now converted this whole thiourea unit from the previous molecule into this guanidine unit. Resulting compound was way less toxic, right, and it was also more potent and more active in comparison to methiamide. The other things about cimetidine was it was metabolically stable, which means when we talk about some drugs being metabolically stable, what do we mean? Every drug, when it is ingested, it goes through liver, right, and liver has certain hydrolyzing enzymes. It is a machine of a plethora of hydrolyzing enzymes, which tends to I told you in the last lecture, we tends to get rid of this drug molecule, considering it as a foreign particle. So, if a drug is metabolically stable, it means it is hard for those liver enzymes to then get rid of this molecule. So, it stays in the blood for a longer period of time. Okay. So, then it is excreted also largely unchanged. The other bad thing about this compound is that it is found to inhibit P450 cytochrome oxidase systems in liver. So, this P450 cytochrome oxidase is a cluster of enzymes which are hydrolytic enzymes, contain all these kind of oxidation reduction reactions in the liver for metabolizing action. Which means that if the problem is that if you have other drugs in conjunction with this particular drug cimetidine, you need to be very careful, and especially if those drugs belong to diazepam, lidocaine, warfarin or theophylline, one should be extremely careful taking cimetidine, because then these molecules, their profile, their therapeutic profile will also change, because this P450 will not be able to act upon these drugs as effectively as anticipated. And then came ranitidine. In this basic scaffold has also been changed to a furan. Other things have remained, this has remained, and here you have an imidine unit this time. So, ranitidine has been a drug with even fewer side effects. It would last longer and was perceived to be 10 times more active than cimetidine. Also importantly for us, it would not inhibit P450 cytochrome oxidase system. There are other drugs also which fits into this H2B receptor regime, but this does not fall into purview of this lecture. We will skipping that here. With this, we next come to neurologically active drugs. And when we talk of neurologically active drugs, what we mean is the drugs which are able to actually cross effectively blood-brain barrier. Okay. So, we really, when we design a drug, unless it is for psychoactive purposes, we do not want that drug to unnecessarily cross a mesh of capillaries at the blood brain junction, so that it does not actually enter brain unnecessarily. But still there are certain drugs which are able to cross this blood brain barrier, and if they happen to cross this blood brain barrier, they tend to be generally psychoactive drugs. So, they would have all the kind of sometimes bad effects of psychoactive drugs. For example, they are addictive drugs, sometimes they create some kind of euphoric reactions and all that stuff. So, analgesics which we are going to study and tranquilizers which we are going to study, they would fall into this category of neurologically active drugs for today's discussion. In case of analgesics, we are going to study a molecule called morphine, and I think you all know about morphine. It is a natural compound obtained from opium, those opium buds, those poppy buds. And I think I told about this molecule in the first lecture itself. It is one of the most effective painkillers available to medicines. I told you that heroin, which was a simply modification of this morphine, was even more effective, but then it was hugely psychoactive. It had those tremendous bad effects, and then it was removed from the market. Morphine acts in the brain and appear to work by elevating the pain threshold, thus decreasing brain's awareness, brain's awareness of pain. Problem with morphine is certain side effects, and what we are more concerned with is are these side effects like excitation, euphoria, and being addictive. And I told you that all of them are because this molecule is very effectively is able to pass this blood brain barrier and then create all these bad effects. Now, in order to reduce the side effects, we certainly need to synthesize morphine analogues to improve activity and to reduce side effects. In order to do that, again like we did in the previous case, it is important for us to understand interaction of morphine with its receptors. The receptor with which it binds are called, again it is a class of receptors called opioid receptors. We are not delving into the details of this receptor here. What I will do is this kind of cartoonish presentation of the active site of that receptor and how morphine binds and fits in that cavity. There are few things to note here. First thing is this aromatic ring in morphine, this ring I am talking about is oriented properly with respect to nitrogen. So, you see nitrogen on one end and this aromatic ring on the other end, and you see that this aromatic ring engages through van der Waals interactions with this hydrophobic binding site. Okay. The second thing is this phenol functionality. So, this phenol functionality engages through a hydrogen bond with this active site here. The third thing is this protonated amine here. So, this protonated amine engages through an ionic bond with again certain residues in the binding site. In addition to all that, you see that there is a huge space, this hollow, this cleft, and this cleft is more or less like hydrophobic cleft, right. So, what you notice here is that in this cleft you have on one extreme this protonated amine, it means an ionic compound, and this ionic compound is joined to this aromatic ring through a 2 carbon bridge. If you consider this 1 carbon and 2 carbon bridge, and this aromatic ring also contains again this polar residue or polar functionality in hydroxy, in phenolic in this case here. So, that should primarily be the pharmacophore or the active molecule, okay. That is the must in order for any molecule to fit into this cavity. So, keeping this in mind, we know that aromatic ring, phenol and nitrogen group in along with that 2 carbon bridge are actually essential for its analgesic property. So, based upon this morphine molecule was simplified a great deal, and people came out with different type of compounds which showed very good activity, very good analgesic activity. For example, this compound shown on the screen, benzomorphins, Likewise, this phenazocine or brimazocine, and in all these cases you see that the structures are certainly much more simplified than morphine, and this pharmacophore has remained intact. So, you see this nitrogen, you see this 2 carbon bridge here, you see this aromatic ring, and you see a phenolic compound, a phenolic functionality here. As far as phenazocine is concerned, it is 4 times more potent than morphine. It has a long term analgesic action, and it is very low as far as addiction risk is concerned. The other compound is 200 times more potent than morphine. It is also non-addictive, and then the other drawbacks associated with morphine are also subdued in this compound. Coming to tranquilizers. So, tranquilizers are actually class of drugs which are used for the treatment of stress, for the treatment of mild or sometimes severe mental diseases. Okay. You note all the components of tranquilizers, all those active molecules which behave as tranquilizers, you see those molecules as essential components of sleep inducing drugs. Okay. Now, these tranquilizers have been found to act again on a receptor, and this receptor is called GABA receptor. GABA stands for gamma aminobutyric acid receptor. So, basically it receives this gamma aminobutyric acid, and hence this name has been given to this receptor. This receptor actually houses, or man keep this ion channel, and this ion channel is primarily for chloride ions. So, it allows this chloride ions to go inside the cell. Okay. That is primarily the job of this particular receptor. Now, if you look at this receptor here in more perspective, you note that this receptor has binding sites for different kind of molecules, one being its natural agonist, okay, and its natural agonist is actually GABA. So, it has a binding site for GABA. It also has binding sites for the drugs which have been used as agonists for this GABA receptor. So, those drugs primarily, and what we are going to study, are barbiturates or benzodiazepines. Barbiturates because they are a class of barbituric acids, and benzodiazepines, again this is the structure of a benzodiazepine. Okay. This is a diazepine, and that is a benzene ring. So, you see receptors, it has more than one receptor, and all those receptors actually do the same job. They are agonistic in function. It means they help to make this flow of chloride bigger and bigger inside the cell. So, let us look at this mode of action in a little bit of details here. So, in the first picture shown here, you have an empty receptor, which means there is no agonist, natural or man-made. If this is the case, this ion channel is blocked, it is stopped. It stops an entry of chloride ion into the cell here, right. As soon as you see in the second picture, its natural agonist GABA binds to this receptor, this cavity, this ion channel is opened, the gates are opened, which causes chloride ion channels, which causes this chloride to enter the cell and hyperpolarize the cell. Okay. Now, let us say you have given an drug molecule. So, then binding of GABA and this drug to the receptor, it results to even further opening of the channel, and it increases the amount of chloride ions going inside. What that mean to us? Now, that would mean to us is entry of chloride ions, and I would say excessive entry of chloride ions. It would hyperpolarize the cell. Okay. So, once it gets hyperpolarized, it becomes very difficult for this cell to depolarize again, and hence it is unable to send the neural excitable or exciting signals. You probably understand the neuron chemistry, wherein this polarization and depolarization of the cells are actually able to provide in exciting signals across the neurons, and that is how the signal is transferred. So, basically through hyperpolarization you have blocked this polarization of the cell, and hence the signal will stop, and physiologically that would mean that a person will start to feel calm and calm. So, the effects would be that a person would feel that soothing and calming effect and probably will go to sleep. Okay. So, basically in this screen now we have the drugs, tranquilizing drugs, and they belong to the two classes which I have already shown. So, you see diazepam here, you see alprazolam here or oxazepam here. The difference is in the minor change in this diazepam structure, like here you have another cyclic heterocyclic structure in addition to these two diazepam structures here. Likewise, in barbiturates one would have veronal, one would have amobarbital and also pentobarbital. Pentobarbital has legendary stories during Second World War, when it was used as a tranquilizer given to those patients, those warriors who were suffering from wounds. Okay, and it is said that it killed more people than saved people, because people that time, physicians really did not know the dose of these barbiturates. So, invariably the patients were fed on more diet of barbiturates, and that has its lethal effects. Okay. Well, I just digressed. So, I will come back to the topic, and now we will study antimicrobials. Antimicrobials are those chemicals which prevent development or inhibit pathogenic action of microbes. These microbes could be bacteria, fungi, virus or any other parasites. So, when we talk of antimicrobial drugs, we talk of antibiotics, we talk of antiseptics, and we talk of disinfectants, and in this lecture we will study all the three. So, let us begin with antibiotics. These are the chemical substances which in low concentrations they inhibit the growth or they destroy bacteria. Okay. As far as mechanism of action of these antibiotics is concerned, we need to first understand a bacterial cell. So, this is a diagram of a bacterial cell. What you note here is there is this thick cell wall in addition to the plasma membrane here. All of us, all human beings and all eukaryotes, we do not have this cell wall, we only have this cell membrane, right. So, it contains this thick membrane here. And then what you note here is some scattered clusters of protein particles required for its various routine functions. Then you also see this cluster here, which is actually those nucleic acids which are its genetic material, and then you have this whole all of these presents in this liquid matrix, which is cytoplasm. So, over a period of time, the kind of antibiotics we have discovered basically fall in 2 to 3 categories. There is this class of antibiotics which actually act upon the cytoplasm, which means it acts on the metabolism of the cell, and the class of compounds which actually does that are sulfonamides. We will be studying them in detail. The other class of compounds or the drugs are the ones which actually work on the cell wall, which act on the cell wall. So, in general it ruptures the cell wall and lyses the cell. The molecules which do that are like penicillin, cephalosporin or cycloserine. We will also discuss penicillin in detail. Then there are drugs which actually act upon those protein particles and those protein machineries, and the drugs falling in this category are chloramphenicol or streptomycins or tetracyclines. Or then there are other drugs which can also act on cellular membranes like polymyxins and others. So, with this kind of mode of action of antibacterial drugs, what we are going to study in this lecture are the drugs which inhibit the cell metabolism, which means it acts on the cytoplasm, and other drugs which inhibit bacterial cell wall synthesis. So, we will be discussing sulfonamides and we will be discussing penicillins. All right. So, let us begin. Discuss about sulfonamides. So, as I said, these are antibiotic agents or antibacterial agents which inhibit cell metabolism. These are also called antimetabolites. The class of compounds I am talking about are sulfonamides. So, you see this benzene structure, this is your sulfonamide moiety here, right. Now, we should understand what kind of metabolism they actually act upon. So, I told you they act on a certain metabolism, that is why we call them antimetabolites, right. So, look at the metabolism, look at that particular process on which these sulfonamides react or act. These sulfonamides are competitive inhibitors of dihydroterate synthetase enzyme. Now, we should understand where this enzyme is used and what is the implication of its dysfunction. This enzyme is required to synthesize a very important vitamin even for us humans, and that vitamin is folic acid, vitamin B9. Folic acid is in a crucial step, in a very crucial step of its synthesis is where you require pteridine, you require para-aminobenzoic acid and also glutamic acid, and all the three will mix together in this enzyme which this sulfonamide inhibits. So, this enzyme is used as a catalyst and this addition reaction takes place which forms folic acid. Sulfonamide, being very similar to this para-aminobenzoic acid in structure, it acts as a competitive inhibitor, and I hope you can recall your knowledge from my last lecture where I spoke at length about competitive inhibition in case of enzymes. So, this sulfonamide acts as a competitive inhibitor of this particular step of this particular enzyme, and once this happens, this enzyme is not able to form this folic acid, and this is required in its cellular growth, in the synthesis of its nuclear genetic material. So, that does not happen and then the cell would die. Now, interesting question which should come to your mind is, I told you this is a vitamin which we also require. The advantage for us is that we take it from plants and other sources. We don't have any machinery within us which synthesizes this, hence we do not have this enzyme. Therefore, any molecule which will fit into this enzyme will not affect us, right, it will only affect the pathogen. That is important. Okay. So, looking at other properties of sulfonamides, they are active against gram-positive bacteria like pneumococcus and meningococcus. They are ineffective against infections such as salmonella driven infections like typhoid. There are other problems with this first line of sulfonamide drugs which was countered, and that problem was during its journey in the blood. This sulfonamide tends to get acylated, and this acylated compound is actually insoluble in water. So, insoluble in blood also, and then that happens to block the kidney tubules. Okay. Now, the other problem why this happens is because of the acidity of this particular hydrogen attached to this nitrogen here. So, what happens is, since this particular structure, this thiazole here, this hydrogen is not very acidic, right, and since it is not very acidic, it remains in an unionized form at the normal blood pH, and that is why as soon as this nitrogen get acylated, this whole molecule tends to get precipitated in the blood. Okay. So, what would be the solution around it? The solution would be to probably enhance acidity of this hydrogen. Okay. And one way to go about it is add more deactivated or electron withdrawing systems. So, instead of a thiazole here, we would replace it with this pyrimidine unit. Okay. And what it does is, because of having two electronegative nitrogens in the vicinity, it tends to impart its electron withdrawing impact and making this hydrogen more acidic. So, now this compound, even if it is acylated, it remains soluble in blood. Why? It remains soluble in blood because I told you its acidity has increased. So, because its acidity has increased, its pKa value has decreased, and now it will remain in this ionized form under normal blood pH, just like a mild acidic compound. and that is what we want. If it remains in an ionized form, it is never going to precipitate in blood. Okay? It will remain there in solution. So, this compound is much more soluble and way less toxic, this sulfadiazine. That is the second line of these sulfur drugs, and likewise you would have other sulfonamide molecules as well. So, next we come to the other class of antibiotics which work through inhibition of cell wall synthesis. Okay. And I told you these are the drug molecules like penicillin or cephalosporins and others. There are certain trademark features of these kind of molecules, the most primary structure being presence of this beta-lactam ring here. Okay. So, beta-lactam, this is an alpha position, nitrogen you count in this fashion. So, from nitrogen you will count, this is alpha, this is beta, and hence you found this beta-lactam ring here. Now, again this will not impact animals or humans, it will only impact bacteria because we do not have cell wall. Right. So, we remained unaffected by penicillin or cephalosporin. So, less side effects. As far as discovery of penicillin is concerned, I hope all of you are aware it is one of those most serendipitous experiments ever conducted in the history of mankind. You probably are aware Alexander Fleming was the discoverer of this molecule, and it was out of serendipity. He left his petri dish in which he was growing Staphylococcus bacteria. He left it, went for a vacation, and during this while there were spores of a mold from penicillin which actually somehow fell on this petri dish, and then they started their antibiotic action. And when this guy returned back and he was washing his petri dishes, he realized that this culture is almost gone. So, that is how it clicked his mind and a revolutionary molecule came into picture. But this was done in 1928, still it took another 12 years till 1940 when another scientists, they were able to isolate this molecule. And the first such penicillin molecule to be isolated was penicillin G. Advantages and certain features of this molecule is it is active against gram positive bacteria. It is a non-toxic molecule, but there are problems. The first problem is it is ineffective when taken orally. So, it has to be taken through intravenous injections. Second is it has a narrow spectrum of activity, and the last one is there are certain bacterial strains which become resistant to this particular penicillin, and they become resistant due to having this particular enzyme in them which is beta lactamase. So, it is very sensitive to this enzyme. Okay. So, let us now see how to get around these problems. So, to overcome these advantages, we must synthesize certain penicillin analogues, right, and in order to do that we must do certain structure activity studies of penicillin. So, we must figure out what features of penicillin are important for activity, and by saying it in other words, we need to describe the pharmacophore which penicillin presents. Now, what we see is in this penicillin molecule that you got to have this lactam ring. I told you this beta lactam ring is very essential. Then you must also have a amide bond through this 6th carbon. Okay. And at this 5th and 6th positions of carbon, one should have a cis stereochemistry of these two hydrogens, and also at this carbon number 3 one should have a carboxylic or free acid present. So, all these are essential features, all these actually constitute the required pharmacophore for this structure. It should be a bicyclic system also. So, now keeping this pharmacophore in context, let us try to alleviate some of the problems we just figured out. And one problem I told you that it needed to be taken through intravenous injections, because this beta lactam compound is highly sensitive to acids. Okay, and that is quite understandable because this is a 4-membered ring, it is a strenous ring. So, as soon as there is even a catalytic amount of acid, this ring will like to fall apart and will like to open up in this manner. So, this is a hydrolytic reaction in the presence of an acid. There is another way also of opening up of this lactam link, and that is through neighboring group participation of this acyl group here. Okay. So, this acyl group can also donate a pair of electrons to this carbon, and in the presence of an acid, this will actually force this molecule, this lactam ring, to fall apart. Now, in order for this to happen, your oxygen should be high, sufficiently electron rich. Okay. So, electron density should be quite high on this oxygen, right. So, we cannot do much about this aspect, the first aspect, but we can certainly try to modify a compound in context of this second problem, that is the neighboring group participation, which is resulting in falling apart of this ring. What we can do? Well, as a synthetic chemist, what you can think of is you can add an electron withdrawing group to this carbonyl compound, to this structure here, right. So, having an electron withdrawing group here will actually migrate electron density towards this end, rather than kind of pumping electrons on this oxygen. Now, if you do that and oxygen is deprived of of electron density, it is very difficult for this oxygen to participate in the kind of cyclization reaction as we saw in the previous slide and opening up of this lactam ring. So, based upon this fact, certain molecules came up in market like oxacillin, like penicillin 5, like ampicillin, and if you notice all these compounds, you would realize there is an intervention of one or more one or more electronegative heteroatom here, be it oxygen, be it nitrogen, or others in this case. Okay? And in all these cases, they were found more acid stable components, more acid stable analogues in comparison to penicillin. Now, coming to the second problem I mentioned, and that happens with the penicillin resistant bacteria, which contains this enzyme beta lactamase, and this enzyme again act on this beta lactam ring, rips apart this linkage here, this lactam linkage here, and opens up the ring. Okay. Now, again going by our knowledge which we have accrued so far, we would probably can think of making certain changes, certain minor changes. So, one such change, keeping all the active residues, keeping all the pharmacophoric units intact. So, one such change we thought of was adding a bulky group here. The scientists decided that they could find an ideal shield in the form of a bulky group, which should be large enough to ward off a lactamase enzyme, which means it should be large enough so that this molecule will no longer fit into the active site of lactamase ring, lactamase enzyme, but it should not be that big, or it should be small enough to allow penicillin to do its duty. It means it should be just large enough to be able to not fit into this lactamase, but it should not be that large that it is not fitting into the cell wall, okay, and do its job, rip apart the cell wall. So, based upon that, other classes of compounds came into picture like oxacillin, like cloxacillin, like this flucloxacillin, and you see in all these structures you would have this additional aromatic ring here, chloro substituted aromatic ring in this case, so that it creates a certain level of steric cloud onto this particular active scaffold. Okay. So, this has been found to be a class of penicillins which were which were also effective on beta lactamase resistant compounds, bacteria. Okay. Now, with this we move to the last topic in hand, and that is about antiseptics and disinfectants. If antibiotics were compounds which were effective against bacteria, antiseptics and disinfectants are compounds which are basically broadly active against several other microorganisms, and they need not really kill the organism, but they can also grow, stop their growth as well. So, we talk of antiseptics which destroy or inhibit growth of microorganisms. Disinfectants, these are chemical agents which are used to inanimate objects or surfaces. certainly disinfectants are more toxic and stronger in case of in comparison to antiseptics. The compound we are going to see here is chlorhexidine, which is an active component of Sablon, widely available in market. It is just 7.5% of this compound here, and this compound can also act as an antiseptic. If you increase this ratio here, the same compound can also behave as disinfectant, okay, by increasing the ratio of this molecule. When we talk of mechanism of action of this particular molecule, as you could note that this molecule is a positively charged compound. You have all these guanidine units which are positively charged. So, these positively charged units in this molecule, they actually come close to negatively charged phosphate groups on the cell wall of a bacteria or other microorganism. Then what happens is, it, because they would kind of attract each other, it finds its way into the cell wall. It is actively transported into the through this cell wall into the cell. There is an interaction between this molecule and phosphate molecules, and once it enters the cell, it actually ruptures the cell wall here, and leakage of these certain inner molecules, for example ATPs and all, will take place and opens up the cell, and cell will die. Okay. So, that is the mode of action of this drug and other antiseptic molecules also. So, with this we are coming close to this third lecture, and I will just briefly revise what we did in today's lecture. We started with antacids. I described functioning of antacids. I spoke about systematic, non-systematic and other antacids and how they behave. Then I moved on to an entirely different class of antacids, which are antihistamines, and I told you, I spoke about there being H2 antagonists and how they are designed and how an effective molecule, cimetidines and others, were designed. Then I moved on to analgesics, and analgesics we studied were of the kind which were psychoactive also, which were able to effectively cross blood brain barrier, and in this case we discussed morphine and its analogues. Then we moved on to tranquilizers, and I spoke about there being GABA inhibitors, and then we discussed some of the molecules, diazepam and barbiturates, as tranquilizers and how they act as agonists instead of antagonists, as we saw in case of analgesics in this particular case. Finally, we came to antibiotics and antiseptics. In case of antibiotics, we studied two different mechanisms of bacterial killing. First was their compounds acting as antimetabolites. In this case, we studied sulfonamides, and in another case we studied how the molecules can actually act on the cell wall, and in this case we studied penicillin. And finally, we studied antiseptics and disinfectants. This lecture was heavily borrowed and prepared from these sources, and I recommend students they can go to these sources to read more about all of these all of the contents of today's lecture. I wish you all the good luck, and thank you very much for staying with me for this third lecture. I hope to see you in the fourth and concluding lecture on this series. Thank you very much.